got the oral cavity, the pharynx, the esophagus, stomach, small intestine, large intestine, rectum. Then we've got all these accessory organs. I'm going to draw on the board in just a couple minutes a picture of how generally you can do this. So we've got two types of digestion, right? We've got mechanical digestion, which is a physical process whereby things are being mashed up or broken down. Your teeth, they perform mechanical digestion. Your tongue, to the extent that it smushes the food up, performs mechanical digestion. The churning action of your stomach is mechanical digestion. It is a physical process. Chemical digestion are reactions which break the bonds within the food. That's where we use the enzymes. We hold the molecule in a certain way so that the bonds can break and they get broken down. To absorb these nutrients, we have a couple of different options, right? You can go paracellular, which means between the cells, or you can go transcellular, which is through the cells. So if you're going, some of the larger molecules can go, or little tiny ones even, can go between two cells, or two cells next to each other, and there's a little channel they can go through. Transcellular means that they're going through the cell itself. They're entering the enterocyte, also known as the small columnar epithelial cell of the small intestine. They're entering that cell, and they have to travel from one side of the cell to the other side of the cell. Most of what we're talking about is going through the cell. When we get to lipids, there are some examples where we're going to talk about paracellular, but we're mostly talking about transcellular. We need to excrete waste products, right? We cannot digest everything that we have. There are some things that are non-digestible, so we have to pass that through. It has endocrine functions, meaning that's cell signaling through hormones, right? So an endocrine gland, the difference between an exocrine and an endocrine. Endocrine gets passed through a duct. Exocrine, oh no, did I just do it backwards? Hold on. I always do that one back. Pause, it's in like slide 6, so I'll tell you in a minute. And immune functions. I always get, there are like 4 things that I always go backwards, and that's one of them. Okay, and immune functions. So the gastrointestinal system functions in an immune capacity, right? There's a lot of research about food allergens and whether or not we're predisposing people to have systemic allergies. They've just reversed, I don't know if you guys saw this, in the last 3 or 4 weeks, there's a brand new study that came out that reversed the recommendations for small infants and peanuts, right? They're finding that they used to say no peanut introduction if there's a risk of allergy, and because there's increased risk of developing the allergy. There's a brand new study that came out and said, nope, we need to be introducing that earlier. We need to follow the clinical recommendations where we work, whether that's a hospital or WIC or wherever we're doing. They're going to take those clinical studies and they're going to put them into practice for their site. So as soon as we think something's fixed in space, it's not. Okay. All right, so the tissue layers of the GI tract. So this is mainly review, right? I'm not going to go. I need to talk all about the lamina propria, and I need you to put in order the circular and the longitudinal muscle. Okay, you did that. But it's here so that you know. We are going to be in detail drawing an enterocyte and use that throughout the thing. So that's different. But all the rest of this is what's in your mucosa, right, which is the top layer. A submucosa is the next layer down. The reason that you learned all this for us is because, so we're getting these nutrients through the enterocyte. How do we get them to the rest of the body? They go through the cell. There's a basolateral membrane. They pass down through there. They get dumped into the bloodstream, and the nutrients circulate around the body. So we're using that base knowledge to assume that you understand that if you're getting dumped into the hepatic portal system, you're eventually ending up in the liver where a lot of our chemical reactions take place. That's what you need this knowledge for. I'm not going to draw the hepatic portal system, okay? So what's the function? Support, blood supply, secretory nerve control. Okay, and then we go to the muscularis externa when we talk about mechanical digestion or moving things through the intestine. These muscles are contracting and moving. If you get the longitudinal and the circular muscle, they oppose each other because you've got to have all these contractions to go back and forth. Or if you've got your stomach, it's not just going like this, it's going like that. Okay, so your adventitia or serosa. Okay, the difference between these two things is adventitia is extraperitoneal. So we have a peritoneum, right? And that's this sort of cavity surrounded by a membrane where most of our organs for the digestive tract are. There are some things that are retroperitoneal or extraperitoneal, like our kidneys. When they say retroperitoneal, that's right, they're in the back. They're outside the peritoneum. That just means that they're not encased in that membrane that makes the peritoneum, which is where most of our digestive organs are. So you can see part of our intestinal tract is outside of the peritoneum. So it's kind of goes back and forth. Okay, so regulation of the gastrointestinal tract. How do we do this? So we've got a nervous system, right? You control most of the motility and the function. And then there are two types, the parasympathetic and the sympathetic. I know this is back of everybody's brains somewhere, right? When we talk more about this, if there's a specific case where I'm talking about it with you, like we're going to talk about alpha-1, 2, and beta-adrenergic receptors later. I'm going to put it in context of this is stimulating the parasympathetic nervous system, and this does these things. But this PowerPoint will be sitting up there, so if you forget what these things do, you can go look. Okay, and we're also later going to learn how we make acetylcholine and how we make norepinephrine. These are things that we use within the digestive tract and without, right? Okay? So, the parasympathetic regulation, parasympathetic, if you think, remember, parasympathetic slows down, sympathetic speeds up. You want to digest more when you're relaxed. So, parasympathetic increases digestion, it increases absorption, and it increases secretion. Sympathetic regulation slows down, it freezes digestion, it freezes absorption, and it freezes secretion. This is because if you are sympathetically activated, you need to be spending your energy somewhere else. Digestion and absorption take energy. And so you need to be in a state where that makes sense for your body to divert energy and resources toward that. It's kind of the mini fight or flight, right? If you've got something else that's really stressing you out, you're not really wanting to put energy toward that. There's a lot of research, have you guys seen those commercials, the cortisol connection, you know, they try to sell you that little thing. So that is a non-FDA approved or regulated extension of this general idea. That if you sympathetically activate yourself for too long, you get the same type of insulin resistance to these types of hormones, and then you go out of whack digestively. I'm not saying that's what actually happens or that supplement works. I'm saying that is what the commercial is basing that on. So it's a lot more nuanced than that, and it's a lot less well understood. Yes?
You said sympathetic slow